the second Indo-US
Nisha, if you can.
PITCH4RARE contest. I am Harsha Rajasimha. I am the founder and executive chair of Indo US Organization for Rare Diseases. I'm excited and grateful to Amgen for supporting and sponsoring this event. We will have a quick introduction to PITCH4RARE, and we'll introduce the judging panel that we have for today's event, and introduction to the pitching companies. We will go through the actual pitches by 6 innovators that will be pitching today. The judges will be scoring each pitch, and we welcome and invite all the attendees to participate in scoring or providing feedback. In the chat, you can start introducing yourself in the chat window throughout the event, and do some networking and also help answer questions or help each other out. And the winners will be announced in a press release in the next few days, and we will be in touch to communicate that and circulate the draft to the pitching companies and judges before that goes out. So Indo US Organization was founded to build bridges between nations, cross-border collaborations, as a lot of innovation and progress in rare diseases has happened, starting with from United States since 1983 Orphan Drug Act. And since then, we have over 1,100 approved orphan drugs, and a lot of these drugs are not accessible in the rest of the world as much yet. And even within the US, it's not affordable to many patients and families. And so there is a lot of work to be done. And these approved drugs only address 5% of the known rare diseases. So 95% of the rare diseases still do not have any approved treatments. And we believe bringing countries like US, India, and others into cross-border collaborations can speed up this process. And in that regard, we have various programs that we host at Indo US Rare, all centered around patients and families affected by rare diseases. Whether it's a patient concierge that provides free care navigation service to patients affected, or patient alliance program with nonprofit foundations advocating for specific rare diseases, education awareness and research programs, and grants funded programs, as well as corporate alliance to engage industry and in advocating to the US Congress, Senate, but also in India and other countries as well, to make sure we remove barriers and overcome these barriers to speed up this process of drug development. So we had our inaugural PITCH4RARE last year. The Indo-US Bridging Rare Summit is organized 1 year in the United States in Washington, DC area, another year in India in the New Delhi area. And so in the true spirit of Indo-US. So last year we conducted this, there were 7 pitches, and the judges selected 2 winners, CRISPR Bits and Keiteki Innovations. And so that set the stage for continuing this contest where innovators, sometimes patients themselves, sometimes it's a family member who gets inspired by their lived experience and make it a life's mission to bring a product to market to help patients live better, quality much longer. And so that's a significant effort, and all of us can help innovators like that. And that's why we continue to build on this PITCH4RARE contest momentum. And we will hear 6 pitchers today and many more to come in the following pitches as well. So the idea is to increase global outreach, to pitch on an online forum. You get to hear feedback from the judging panel itself, but also other collaborators. And beyond this event, we will distribute this pitch recordings globally on social media, and also announce on press releases and so on. So you'll get more visibility about your story and what you're building, and have better impact, networking and connections. You need to meet that one person that can make a difference in your journey, and maybe many more. So that's where this could set the stage for that. Everybody who pitches today is a winner, but the judging panel may select one or more winners, and those will get some extra benefit, visibility, attention, maybe in-kind support and mentoring, and coaching and connections, warm introductions, not just by the judging panel who out of their goodwill and heart they might do that, but also many people who are attending this call right now, as well as others who will learn about your story as we share this more widely. So with that, let's do a quick round of introduction of the judging panel. So we are grateful to this eminent panel of judges who have kindly agreed to spare their time and share their feedback today. So Abhishek Mathur, if you could give a brief introduction about yourself.
Hello, everyone. This is great to be here for PITCH4RARE contest, and thanks, Harsha, for bringing me into the panel of judges. So, background-wise, 20 years into industry, and I started my career with Amgen in US in the CMC space, CMC development space. Worked for Regeneron after that. Along the way, did my MBA from Duke University, and moved to India in 2018 through Amgen again in the discovery space. And I worked with a couple of folks after I moved to India. I can see a few names, but overall a CMC person around 20 years of industry. But I'm a chemical engineer, but did my PhD in biological sciences. So glad to be here and looking forward to some exciting pitches.
Thank you, Abhishek. Chidi, if you're on the call, please unmute yourself and introduce yourself.
Hey everyone, my name is Chidiebube. I am the venture studio manager at ETC Baltimore, and I help with due diligence. I help with making investment decisions, and I just totally help companies in the Baltimore region to be able to just have successes, be able to get the resources that they need. And we have a venture studio where we focus on biotech, medtech companies. And so I'm really glad to be here and be able to get to know the new companies that I'm not aware of. Most of the companies I haven't heard of, so I'm really interested to hear and see what their ideas and what their companies are about.
Thank you, Chidi. Thanks for joining. And Christiaan Engstrom from Bullpen Club.
Hi, Christiaan Engstrom. It's actually just BLPN, no club.
Okay.
Yep, and that's okay. We went through a branding change, so everybody is having a hard time with it. So I accept responsibility for the confusion. I run a group of life science deal makers is the best way to explain it. And every time I try to explain what we do, I get caught up and it's better experience. We have a website. It's blpn.club if you want to go check that out and look at what we're doing. We have a mantra. If you join our club, you're committed to helping each other. So the idea is that we can create, like to say, reverse the energy for folks that are trying to get deals done in the marketplace. We can bring resources to each other, and that's what I've been doing for the last few years, building that. My background, just real quick, I ran a group of pain management clinics in the United States and exited that, sold it to a PE group. I took a phase 3 immunotherapy program on, had an orphan designation, and unfortunately kind of took that when it was headed towards bankruptcy, spun out a contract manufacturer of antibodies and served various industries, cell therapy, immunotherapy, research, diagnostics, and then moved on, exited that, and then took on a spin out cell therapy cryopreservation solution. I am not a scientist, so every time I take on one of these deals, I have to learn some of the terminology and try to bring the business discipline to it. So today, when I'm listening to you, I'm going to be thinking about your pitch and thinking about how it might be heard by investors and giving my feedback kind of based upon that. Thanks, Harsha.
Thank you, Christiaan, for joining us. And I'm personally inspired by Christiaan's effort in bringing this mantra of find someone, help, rinse and repeat. And so this Pitch for Rare contest is actually inspired by that same mantra. So no single person or judge here is necessarily an expert in all of rare diseases. But one thing that's common is everybody wants to help and contribute in whichever way they can. And that's the mantra that we will lead with. So Mahesh Narayanan from Match Play Group.
Thank you, Harsha. Pleasure to be here, and looking forward to all the pitches from all these amazing companies as well. Thank you for inviting me to be a judge in this event. My background is primarily in life sciences. I've been in the industry for a little over 20 years, both as a founder and now on the investment side. My background's primarily in neurology. Did my graduate work at Penn. Worked in multiple pharmaceutical companies, including GSK, as part of their clinical neuro team, and then also did a small stunt as an adjunct professor of anatomy and physiology at Philadelphia University. But since then, I've moved on to be the managing partner at Innovation Ventures, which is an early stage neurotech firm, also based in Baltimore. But I'm also the CEO of Match Play Group, which is a family of funds that primarily invest in life sciences and fintech. And we also partner with multiple other funds for helping them with due diligence as well as deal flow. And looking forward to hearing all the companies pitch today. Thank you.
Thank you, Mahesh. It's fantastic to be part of the Match Play Group's event last month in New York City, and fantastic ecosystem that you are also building like Bullpen and others as well. So great to be part of it, and thanks for joining today. Narendra Chirmule, are you here today? I know you had a conflict. Probably not. So he's unable. That's why he referred Abhishek Mathur. So grateful to Narendra for supporting this. Dr. Mandeep Kaur.
Hello, everybody. Hey, Harsha. Thank you for the kind invite here as well, Mandeep Kaur. I'm a physician executive. I've been, after finishing my fellowship at Wake Forest University School of Medicine in North Carolina, I was a faculty there, and then I moved from academia, from a hospital setting to biotech pharma, because I was really excited about how the clinical research was moving forward, but also about innovation. So I've been in the industry, be it a small pharma like Arena Therapeutics or big pharma like Sanofi, Pfizer, Amgen for about 20 plus years now. Done many, many launches. I think the last time I checked, about 19 launches and about 4 M&A mergers and acquisition deals. Something that I really enjoy is innovation, as that's been my career journey. And I'm so looking forward to hearing some great ideas and how you guys are preparing your journey in this phase as well, and here to help you and support you. So thank you again.
Thank you, Mandeep. So all highly accomplished experts in their own fields, and some generalists, some specialists, but I'm excited to have all of you, and thank you so much for generously providing your time today. So let's move to, we did have biograph slides. Sorry, I was supposed to flash them while you were introducing. So we'll get through individual bios of the judging panel, which you heard from each one of them. So thank you all. And Naren is a huge supporter of both our summit, and he is a professor at Indian Institute of Technology, and goes between Pennsylvania and India frequently, and great champion for scientists and entrepreneurs. So even though he's not here with us in person today. So with that, let's jump into the pitches. We have 6 pitches lined up. Is that correct, Ramya?
Yes, yes, Harsha.
Okay. So
we have 6 pitches.
Yes. So thanks to Ramya primarily and Nisha for organizing this whole event. And so the way this is going to go now is each company will pitch for 5 to 7 minutes. And the remaining 3 to 5 minutes is for question and answers. The judging panel will ask questions live on the screen after the pitches. Everybody else is welcome to put their questions or comments in the chat window, but please refrain from unmuting yourself. You're welcome to stay and listen to all the pitches and the interactions, but it's the judging panel that will have interactions with the pitchers. So each company will pitch for 5 to 7 minutes, and the remaining time for question and answers. My suggestion is keep your responses as brief and to the point as possible, so you can take 3 to 5 questions in that short period of time, like 30-second, maximum 60-second responses to questions. And in the pitches, emphasize clearly on exactly what problem you are addressing, what is your solution, what's your unique differentiation or competitive advantage, and your sustainability plan. As we recognize in rare disease, a lot of this is mission driven, but at the same time that has to be sustainable. And finally, do include your ask on your closing slide so we know how we can help you. What are the areas where you are seeking help as well? Any questions before we jump into the pitches itself? And the judging panel should have a scoring sheet. Please confirm if you have the scoring sheet. Okay, great. I see a thumbs up there. Okay, great. And so here are the 6 pitchers today. And this will be the sequence of pitching. So 10 minutes per pitch, including question and answers. We will take 1 minute after each pitch for the judges to complete their scoring, and then we'll move to the next pitch. And as we can see, a good mix of pitchers from US and India, and it's a great diverse judging panel as well, which just naturally developed that way. So I think this is going to be fantastic hearing each of these pitches, what problem and how they are going to solve this. Are we ready to get started? Okay, so I will turn it over to TL Srinath from Genofi Biotech Private Limited. Srinath, let me know when you're ready and we can start your clock.
Sure. Thanks a lot. Am I audible?
Yes, you are.
Yeah, kindly permit to share this screen. Done. Yes. All right. At the onset, I want to thank one and all because this is a rare event where, it's not an ask for money. Basically, it is an ask for networking and try looking out how to help each other in this direction for the wonderful cause of rare disease people. All right, so this is the elevator pitch to start with. Chuckles is a first of its kind prescription nutraceutical being developed for epilepsy management, suitable for individuals with any form of epilepsy. So that includes people with rare epileptic disease also. By 2031, we want to capture 3.5% of the 16.9 million epilepsy market in a fun and stigma-free format. As you can see, it's a sachet with a dissolvable material being there. At Genofi, we keep people at the center of our heart, so because they are all phenomenal, and our science will help them live that way and every day. And we are supported and associated with CCAMP, BIRAC, as well as Research Cell of IIM Bangalore. This is the team. I'm Srinath, and I bring transform science into innovation and tangible products. Geetika is COO who brings in the diverse experience of the industry into the operations and management. Tejas, who has done postdoc from HHMI, and he brings his protein biology into the action. And Professor Upendra, who is a distinguished scientist from Indian Institute of Science, it is his IP that we have exclusively in license and taking it forward. Now, epilepsy is a chronic neurological disorder, and it is not a static kind of thing. As it progresses, the severity also increases. And it happens to be the most common disorder prevailing in 50 million people worldwide. The burden is not just the medical, but it is social as well as economic on the whole family as per se. And neuropsychiatric and cognitive impairment is the comorbidity. So what it leads, there is a requirement of a medication. Yeah, there are a number of medications there, but there are 15 million people who are non-responders, and those who are turned cured, some of the time, like 30% of them relapse. And we all know most of the medications have some kind of issues which can impact the organs or the function of the person. And most of the drugs of epilepsy are actually symptomatic by nature. And the medical food which is offered has a limited control, like ketogenic diet. Well, who has gone into dieting, they know that very well that the palate is not that great and it leads to a lot many gastrointestinal issues. And there is a caregiver burden as well. So when severity is more, the doctors actually suggest for going surgery, which has a variable outcome. Now here is the opportunity gap. That is, mitochondrial dysfunction is linked with seizure susceptibility as well as the epileptic progression. And this is where our product, which is Chuckles, will redefine the epilepsy management. Because here the mitochondrial health is restored through urolithin A, which is a US FDA GRAS status molecule. And then you can clearly see it's a caregiver-friendly format where actually a household person need not to worry about creating something. And it is something which can be carried in a pocket or a bag. So all the stakeholders are essentially happy with that. That's what we hoped it to be. So science is that when there is a trigger, there is a mitochondrial dysfunction, which leads to abnormal activity and leads to seizure. Now here, once a person has been called as that you have epilepsy by disease, then if the person is taking Chuckles, which has urolithin A, and sustainably in every A, it circulates in the body, has a regulation of the mitochondrial function. And let's say if a trigger happens, it may have a lesser severity or may not actually, because we have to prove that point. But eventually, a person will be able to mitigate their whole life process, and that's a gain. Now, here's the competition in the market. Let's say in the business world, we will create a new market segment which is for epilepsy management and we will be having a first movers advantage because it's a prescription format nutraceutical and it is IP protected, which we have filed both in US as well as EU. It has the FDA GRAS status compounds in it, and this is a sticky format for the consumer. And you can clearly see in comparison to others which are listed here, most of them are not present in the Indian market. So this gives us a very big scope to emerge as a player in the market in India. Now, this is the population demography. You can clearly see by in 2021, there were 51.7 million people affected. And there is like 25 to 30% of each adult and pediatric are drug resistant by genetic method or whichever reason behind it. And we really want to capture the market with this product around serving 2 million people worldwide and have something like $592 million in revenue. Because of the IP part, we will be able to expand from the Indian geography further. So this is the revenue model. So when we in-license from ISC, we have got the rights to sub-license again, and then we can happily go into co-branding. And if we can have a more production in other regions, we will be able to do that. And the product sales will be either through the prescription, as I said, at a pharmacy, or it can be a door delivery. And not only that, there are actually many rehabilitation centers and care centers in India, close to 300 kind of the number, where this can be given to them. So there is a whole way of channels to reach out to the consumers.
5.5 minute warning.
Yeah. So almost done. So we are at TRL3. So we want to do more preclinical trials because till now we have used a rodent, but not really a genetic kind of model. So that's the ask. We want to have access to animal models with rare epilepsy so that we can test out our formulations in that. So this is our 18-month plan where we have to do animal studies as well as we have to take it forward so that we can submit it to FSSAI, which is a regulatory body in India for food for special medical purpose. So that is a prescription nutraceutical. So it has to go through the prescription alone. I forgot to mention that one of the hospital in Bangalore, which is pediatric nutrition, pediatrician has agreed to have a clinical trial at his place. And I am in discussion with 2 hospitals in Hyderabad. Yeah, that's it. I am Srinath, open for answering any questions.
Great job, Srinath. 6 minutes, 25 seconds, timing wise. Time for questions. Go ahead, Abhishek.
Srinath, I think well presented pitch. Quick question I have is, what is the biological mechanism at molecular level that would justify the nutraceutical effect? What in vitro or in vivo data you have at this point?
Okay, so here is the slide. So this is actually, they have submitted for publication and not yet done, but they have put into the open kind of source. If someone is interested, have a look at this scientific publication for science people who really want to understand things. All right. So when Upendra's lab did the work, they worked with Drosophila larvae mutant. Actually, you can see DR mutant means it's a drug-resistant mutant. So when urolithin was given to it, you can actually see new butons coming over here. So that means neuronal growth has been promoted. That's the basic thing which they have understood there. So second thing was that how it works in a chronic animal model. So here it is that they had a time plan of feeding the urolithin to it. And when the first time point was there of 60 minutes, in fact, a good amount of urolithin had actually crossed through the blood-brain barrier. And then they did a pull down assay and everything. Through that only they figured out it's a VDAC1. It's a novel target. Right now, there is no drug which is approved in the market which acts on VDAC1. What urolithin A does is, just one more point, is that when the mitochondrial function in case of epilepsy is dysregulated, and it has to go into apoptosis. But here, apoptosis doesn't happen. So urolithin A leads it to bring it to the level that the mitochondria are more better functional. So it induces the apoptosis in it. And remaining, you can clearly see in this graph, when a continuous administration of urolithin had been there, so significantly the seizure score has come down in animal model. Yeah, this is the science behind it.
Okay.
Great.
Thank you.
Great job pitching. I have a question. Have you done any customer discovery to understand if neurologists would want to adapt this into their treatment, or what are their thoughts on your drug?
Yeah. Yeah. So in India, there is the hospital I was referring in Hyderabad. So it is called as, he's a doctor who founded many hospitals in Hyderabad. When I pitched to him, he clearly said that you don't say that this will work for, you are coming it as a solution for people with drug-resistant epilepsy. The way this molecule seems to work is at a fundamental level. So that means you can happily bring out, and we will be willing to take it further if it proves the point. So adoption-wise, yes, because they are the influencers, if you want to say that, because until unless the doctor doesn't prescribe, it won't be given to the patient, or here decision maker is actually a parent as well. Yeah.
We have 30 seconds, two more questions. Christiaan?
Okay, you said you wanted connections. So I would say, to what end, funding seems to be where you need to go. But
Yeah.
I did give you a couple of connections. There's someone in the bullpen called Vasily Kotlov, and he's pursuing mitochondria health with a very similar strategy. He has an over-the-counter drink. That's the first link
Wonderful.
I saved. And then I give you his link. And then there's Metargos, which is an early stage, a seed stage biotech. He's aiming at clinical trials in the US. And then my follow-up questions would be along the lines, I don't understand the India ecosystem, but I do understand Indian companies trying to break into the US market. And I'd have a lot of follow-on questions about your strategy and what sort of research you've done and who you've talked to and how you've socialized it. But Vasily has been doing that for the last 2 years, and he would be able to tell you specifically what he's encountered. Okay, I'll stop.
Wonderful. Thank you. Thank you, Christiaan.
Thank you, Christiaan.
Connect further on LinkedIn. Yeah.
Yeah. Mandeep, a very quick one from you.
Yeah, I think Christiaan has taken all my time, Christiaan. We'll talk about it. No, Christiaan, I agree with you about Vasily, because I was thinking about it. I saw your note in the chat as well. So I think that'll be a good connection for you, Srinath, on that level. I think the question I had was, Srinath, also will be helpful, and this is the clinician in me, because this is coming down to 5-minute pitch here, but it will be helpful to understand the acute seizure versus refractory seizure. Which phases are you looking for in this? Because, like I said, that there is going to be a nutraceutical component because there's a deficiency, there's a weight component, but I think having that little bit of a clarification can also set up you for follow-up conversations accordingly as well. So just more of a comment than a question at this point.
Thank you. Thank you. So
Okay.
refractory is what we were addressing. Yeah. Thank you.
Thank you very much, Srinath.
Yeah.
we're upon time. So judges, please complete your scores for Srinath and Jinofi while we move to the next pitch by Tarun Karthik Kumar Mamidi. So that's the four-word Indian name. So, Tarun, please take it away. Mamidi Health.
Yeah, I blame my parents for such I didn't get to choose.
Go ahead, Tarun. Great to see you again.
All right. Can you all see it? Okay.
Yes, yes.
Awesome. Do you want me to wait until the
No, you're ready to start.
Okay, cool. Awesome. Hello, I'm Tarun. I'm working on AI agents that deliver faster, accurate, and affordable genetic results. Do you know that 20% of 1 in 20 people are affected with a genetic disease, and 80% of rare diseases are genetic in origin? So what happens in this genetic diagnosis process is when you go to a diagnostic center for genetic testing, these are some of the genetic tests that you can get. It takes around 4 to 6 weeks to get a report back with whatever mutations the patient has. So what the diagnostic center does is it collects samples, DNA, sequence the DNA, analyze it to find genetic mutations, and generate the report to send it back to the doctor. So the problem here is per patient, you see anywhere around 5 million mutations. And for diagnostic centers or geneticists to go through these mutations to figure out which mutation is causing the disease takes a really, really long time, and it's a manual process. And within this time frame, the patients and hospitals also spend a lot of money in this diagnostic odyssey. So for diagnostic centers, it's the turnaround time for each patient to sequence their data, analyze it, and send back the reports. For geneticists, there is a vast amount of literature. I mean, every day there are millions of papers coming out, right? So you need to review them and figure out which mutations are causing the disease. And in a lot of cases, you don't know from these disease databases that if a gene of interest is actually causing the disease. So they need to reclassify these mutations based on ACMG criteria and so on. And for patients and families, this is a longer diagnostic odyssey. And for, let's say, for an appointment for a geneticist, you have to wait weeks, right? And what do these families do? They go to Google or ChatGPT to find answers. So our solution is a AI conversational agent that's easy to use and specialized in analyzing genetic data. It reduces time as you can automate this process, and it's easy to integrate into your existing systems. So this is an example of how this platform works. You ask to list out the pathogenic mutations in a patient. It makes a plan. It executes the plan in steps, which is transparent. So it's not a black box, and diagnostic centers can trust. And it returns with a list of mutations. This is an example of how we can work. And we have a bunch of tools helping the agent achieve this. It is also HIPAA compliant. You can detect PHI, and you can safely remove it before you send data to the agent. And you can also connect this agent to your existing systems via MCPs, model context protocols. This is an example of from Claude Desktop asking Ava to look up clinical trials for this variant and a disease. Coming to the competitive advantage, there are other platforms out there that are not customizable. You don't have control of the data. They don't have this agentic layer that you can ask for a second opinion if you have doubts on a mutation or a disease. And they're not affordable. We have released the MVP 2 months ago, and we have almost 35 researchers testing and validating the product. We have 2 pilots running at University of Alabama at Birmingham on a pulmonary arterial hypertension disease, and in Children's Mercy in Kansas City, focusing on leukemias. A revenue model for diagnostic centers, it's a faster turnaround time for research labs. For some of these rare and ultra-rare diseases, they can resolve the misdiagnosis or find new mutations and genes that are causing the disease. And for patients or families, it's a second opinion from this expert AI agent to learn more about the diagnosis. And we have a SaaS subscription for diagnostic centers. It's pricing per sample and enterprise licenses.
5.5 minute warning.
Okay, thanks. Coming to the market size, the genomics data analysis market is growing rapidly with the cost for sequencing coming down a lot. Our TAM is 7 billion, SAM 3 billion, and 2.5 billion for SOM. And I'm Tarun. I have over 10 years of experience in building AI tools for genetic diagnosis, and I'm supported by a lot of organizations, both in India and US. My ask is to connect with mentors, either geneticists or genetic counselors, so we can test the product, connect with diagnostic centers and hospitals, so we can benchmark how much time they can save. And we're also raising a pre-seed round of 100K. Thank you, and I'll take questions.
Excellent. Thank you, Tarun. 6.5 minutes, another 3 minutes. So let's hear from our judges. Who likes to go first? Mandeep?
Go ahead, Abhishek. I think Abhishek went first. Go
Sorry,
ahead,
okay.
Abhishek.
That's fine.
No worries.
It doesn't matter. Okay. Great work for sure. Quick questions around. I have 2 questions. One, what data sources and maybe reference databases does your system integrate, and how are they kept up to date? Right? Do you have continuous access to them in the cloud or something like that? And second is, in a way, what moat prevents any other large genomics player from releasing a similar agentic tool in 6 months? So one is a technical question and one is a more on the business commercial side.
So we have a lot of publicly available databases that are hosted on our cloud platform. Like ClinVar releases their database every other week, I think. So we keep them updated, and since it's an AI agent, it has access to web search and annotation tools. So when you ask something about, hey, what is the updated ClinVar for this? It goes and fetches, hey, this variant is reclassified as pathogenic, like recently by this lab. So that's how we can update classifications and all. For genetic, for let's say other genetic players to build this kind of platform, it seems like a simple agent and a chatbot, but there is a lot that went to developing this, whether it's a model context protocol that's easily integrated with existing systems, or the way the agent, queries and parses the.
Tarun, it's 2 minutes on the single question. So
Sorry.
in the interest of time, let's move to Mandeep.
Sure.
Okay. So I will say, Tarun, on that note, Abhishek has already asked about the data source question, but my question is, how are you defining fast? Because I know there are a lot of other data sources out there too, and different organizations, because yours is fast and accurate. I think I understood the accuracy piece with the data sources and the
Yes.
methodology you're following. How are you defining fast? And then the other aspect is more around, what is your main focus? Because based on the two collaborations, partnerships you have, look like they're different sizes, different indication, different patient population. So what is your key focus as we move forward?
Sure. When I say fast, the agent can, depending on the question, whether it's a simple one or a complex one, the agent can write SQL queries or knowledge graph queries and execute them to get the list, the example I showed you, like really quick. Executing SQL queries in cloud is really fast, right? So we get fast answers depending on the question. Sorry, what's the second question?
The focus area,
Okay.
as your two collaborations are more around different patient populations, but also sizes of the organization.
Yeah, both of them are research labs. So our goal is to find any new diagnosis with the leukemias and hypertension projects.
Okay,
Very
thank
quick
you.
comment from Mahesh, and we are over time.
Thanks, Harsha. I'm happy to just send the email. It's going to be more detailed, I think. So I'll
Okay.
just email it to Tarun, no worries.
Sure. I know Christian has dropped a few connections in chat as well.
I just want to give a quick fire on this for Tarun. If you want to follow up with me, you can follow up with me. And this is going to be a bit harsh, okay?
Sure.
You have a commodity product. It's not special. If you go to Mayo Clinic and see what they're doing in the genomic space, or you go to Moffitt and see what they're doing in the systems that they're building, what is special is execution.
Correct.
It's very hard to sell into labs. So I respect that you now have a couple pilots going. That's really interesting to me as an investor. I have invested in another company, ugenome.ai. You can look at them. They are starting to get revenue through conquest in the marketplace, and that's going to be the race. So that's all that talks in this space in the US, in my opinion, because it's just an idea. Execution is what matters. So then I would say, who on your team has shown execution in this space that can sell into hospital systems? And not just once, but get that contract, that ARR that we're looking for in this space. So if you don't have that person, it's going to be difficult for anyone to believe that you can actually execute on this potential strategy that you have. So I would say focus in those areas. Also, I want you to woo us as a CEO. You were very boring today, and I think you can do better. And I'm just going to give you that. Every opportunity you get to pitch is practice for you. Don't be shy. Everybody's dependent on you. Patients are, your partners, your teammates. You have to sell this. And if you can't, consider someone else maybe within your team that can. And that's straight up. Like, I think actually the CSO or CMO is a more honorable position within the company. You stay longer. You don't have to sell your soul for money. You get to focus on developing the product, right? So just think about that. I didn't mean that to be too personal. Anyways, thanks for giving me a little extra time, Harsha.
Thank you
Thanks
so
for the
much.
feedback.
Great feedback, and Christian is candid and bold enough to give the real feedback you want to hear, Tarun. So great job, and let's move to Brunda Pancholi of Multifly Lifestyle Private Limited. Brunda, please take it away.
Am I audible to all?
Yes, you are.
And the screen is also visible, right?
Yes, it is.
So, good morning and good evening, all. Now, moving from hardcore science and medical sciences based, we are introducing ourselves as a lifestyle product. We make adaptive clothing solutions for patients, elderly, and persons with disabilities. And the unique side of our work is also these clothes are made and marketed by persons with disabilities. So we are a social enterprise based out of Bangalore, India. 2.5 years in operations. The problem we are addressing is the exclusion and loss of dignity faced by many people. For example, in India, there are around 60 million persons with disabilities. There are 10 million persons with chronic ailments, and there are 105 million people living in senior age. So there are enough huge population which is depending on family members, professional caregivers, and feel vulnerable and excluded because of the ill-fitting clothing or unsuitable clothing they have to bear with. And that's why their participation in family, society, and economy is also restricted. At the same time, looking at the other stakeholder, caregivers feel stressed, burdened to constantly care for the person, and they also face health risks. And again, it's a constant loop of dependence, exclusion, and ill health. The solution we provide or create is adaptive design, which may ensure dignity and independence of persons with various conditions. So our clothes have easy fastening options like magnetic buttons or magnetic zippers, flexible fits, uh, tailoring adjustments which can help people on wheelchair sit longer for with urine bag and other conditions. There are discrete adaptive features that make dressing effortless, also restoring their confidence and autonomy. So we believe and we strongly advocate for people to live dignified and active life as far as they can. And with our inclusive clothing, we try to help them with that. This is again, as I mentioned earlier, caregivers have constant strain on them, and by combining our functionality and fashion, we ensure that caregivers are less burdened, and people who are undergoing a health condition feel socially included. They feel like going out in public, start their careers again, and then again slowly it changes the well-being and participation of people affected. What we have unique about us is our fashion is for dignity and adaptive clothing, which are stylish. They are not boring and dull like hospital wear, so to say. They are trendy, fashionable, and functional, to say the least. Definitely, they restore independence and confidence. They also give many people aspiration to dress up better and be part of fashion shows, be part of conferences and meetings and all of that. And the third unique advantage we have is the technical expertise of co-founder Mehul, who is sitting with me here. His experience in fashion industry, the tech know-how about textile to fabric to manufacturing process helps us in quick, fast turnaround. And again, as I said in the beginning, this is fashion by persons with disabilities, but made for all. What we seek from this jury today is around $150,000 for a 2-year runway. We are a small bootstrap startup right now and running on very strict budgets. So this will help us sustain for 2 years. We want to connect with partners and stakeholders of Indo US Care in both the countries. So patient support groups to hospitals to other stakeholders, we want to partner with them so that we can reach out to more, spread awareness to more people. And we also need advice and guidance about rare diseases because it's a new arena for us also. And yeah, how can we ease everyone's life is our motive. I'll show some of the products that we have developed so far. The first one is an innerwear for SMA patient who wants to live independent life. So this has Velcro fittings, which can open comfortably. Again, not adding to the environmental pollution by using diapers. This is a cloth-based washable innerwear. The other one is for people with muscular sclerosis or spina bifida, where the shirt opens from the back side. So the person does not have to move much. The back side also has press buttons to close the shirt. The third one is no friction open magnetic shirt for very sensitive skin challenges. So that's another product. This is a new product that we have developed, shorts for pressure sores. We have realized that many people with spinal cord injury or rare diseases are wheelchair bound for life or longer periods, and they develop pressure sores while remaining seated on wheelchair for long. So this is a garment we have developed which creates compression on the buttocks, helping them reduce anal frictions and fissures.
5.5 minute warning.
Yeah. So these are the fashionable invisible zipper trousers. And the last one, the product which is lying low, is trousers for hemophilia patients, which is inbuilt padding for the knee covering and knee support. And we have seamless socks, again for sensory challenges, and very super fine t-shirts which don't have seams inside. So helping people to wear comfortably. That's it from my side. Thank you for patient listening.
Thank you, Brunda. Great job. Judges. Mandeep.
Yeah, I will say first, Brendan, thank you so much for such an enticing presentation. Definitely a need. My experience in this conversation will be more like I've developed clothing for eczema and blistering disorders. So that's the experience I'm bringing to these for this conversation. I will definitely say from a rare disease, one thing that we have to consider is also the quality of clothing, because they will also have a lot of skin disorders that comes with it, and especially if they've been in one position or in a scenario where they are pressure areas like you were talking about, Brenda, with regard to some of the clothing you shared. I think it'll be very helpful. One part that I was not clear, and maybe I missed it in the presentation, was how long you guys have been doing this and the reason behind doing it, because I think that will also, the story will go a long way if there is a purpose behind it, because that's what I've seen being helpful in other clothing conversations I've been a part of.
So we started it 2.5 years ago. So I am in social sector for around 22 years, and Mehul is in fashion industry for 25 years. Around COVID time, we saw many people, with comorbidities suffering at home. And that time, clothing was also a necessity for people who couldn't get diapers on time, or who couldn't, get clothing washed on time, especially elderly and patients. So that was one of the impetus for us to start, but also my work in a disability space. I've been working with different disability NGOs in Bangalore and across India for last 10 years. And there I saw the need for acute, fine clothing for children with autism and ADHD, to wheelchair users carrying a urine bag and losing dignity while, being out in public. So all those experiences and inputs mixed together made us start this.
Thank you, Brenda. Thank you for sharing.
Thank you.
Mahesh.
Thank you, Harsha, and thank you, Brenda, as well. Great story there. In terms of my understanding is more towards, I guess, the end users, and how do you kind of go the payment model? Because obviously, you have to be you have to make sure it's affordable, but also, to Mandeep's point, you want to make sure there's quality that's maintained. So, who typically pays? What is the current, payment model that you have? Are there subsidies? Is there the grants? Can you just walk me through a little bit more of the economics of how all this works?
Sure. So all our products are priced very reasonably. They are in mid-range of the clothing brands that we get in open market. So basic price like of innerwear starts from ₹500 to the trousers, which are very high-end, very functional, go up to around 2,500 rupees. So that's the range we operate in. We have two models. One is direct to customers, where people get to know about us, or we reach out to them and then they order. Many of them are working professionals or economically well-off people who can afford buying separate clothes, but many cannot afford also. So they can compromise with regular track pants available in the market, which is again polyester and other synthetic material. So we cater to both the spectrums. When it comes to people who can't afford our clothing, we go through CSR route, the corporate social responsibility, where they sponsor for X number of people. There are some social groups like Rotary and Lions and all of them. They also sponsor clothes. And then the third route we take is going to institutions like hospitals, early intervention centers, where there are clients coming for therapy and rehabilitation. We sell products to them and they in turn pass it on to the patients.
And we also try to influence the influencers, the doctors, therapists, specialists, special educators, etc.
Yeah, very quick comment from Abhishek, and we are up on time.
Yeah, so very quick question as well. How did you validate that these needs are unmet by currently available adaptive apparel on the market? And second is it compatible with the medical equipment like catheter supports or prosthetics?
So, what answer?
You have 30 seconds.
So we have customers who are wearing our garments, and these are all customized garments. Okay, so we understand the person's requirement through the users and the caregivers as well, and the influencer or doctors, and then we design. Persons on wheelchair carrying a urine bag, they're using it. They're repeat customers, and that validates our product. Regarding the prosthetists, this has routed through the rehab centers and been very well tried by them. Regarding the medical part, we also ensure that we provide special finishes which takes care of hygiene, like plant-based antimicrobial treatment.
Great. Thank you so much. Let's congratulate Brunda for a fantastic pitch, and let's move on to the next one.
Thank you.
Which is Sugant Murugraj from Atox Research Labs. Sugant, please take it over.
Thank you so much. Hi, I am Sugant Murugraj from Atox Research Lab. We are tackling AMR, antimicrobial resistant. One patient admitted in ICU affected with any UTI or bloodstream infection. In this condition, doctor prescribed medicine based on an culture antibiotic sensitivity test. But there was issue. Currently available all culture-based method give results after 24 hours. But bacteria in patient body replicate every 20 minutes. The timing delay due to diagnostics sometimes leads to patient death. One study released by Lancet in 2023 stated 1.9 million lives in danger in 2050 due to AMR. Another study released by ICMR in 2023 stated that totally 230 people died due to UTI AMR out of 1,000 patients. Another main issue is the antimicrobial resistant pattern differ from region to region. In this condition, we don't criticize our doctors because they don't have access to get region-specific antibiotic sensitivity data. Then, most of our current culture-based method give results after 24 to 48 hours. Then, many of our Tier 2, Tier 3 hospital don't have proper microbiology lab for do AST test. So, we are working on last 2 years. Now we introduced our Bactolizer. It's our Bactolizer. It's a benchtop model. It's our third version. And our working model is first we collect the sample from patient, then we load sample into our antibiotic panel. Then we load antibiotic panel into our Bactolizer system. Wait for a few minutes. We got first result in 30 minutes, fourth result in 120 minutes. We compare first and fourth result until and say the sample is antibiotic resistant or not. It's our test result generated by Bactolizer. We start with Staphylococcus aureus and E. coli. It's our confirmatory study. In this study, we do and analyze MPCC approved 8 culture. The sample have bacteria. You see these kind of long linear graph. But if the sample don't have bacteria, we got linear graph. Based on this graph, we tell the antibiotic effectively kill the bacteria or not. So our product development life cycle, we start with AB1. Currently, we are in AB3. Currently, we do preclinical trial at Karpagam Institute. So value proposition, one hospital have our bacterial no need to purchase of any additional reagents like fluorescence or dyes. And our bacterial is customizable for blood-based analysis, urine-based analysis, effluent sample analysis. And our bacterial we analyzed bacterial based on membrane potential. So our bacterial support wide variety of bacteria and antibiotic, and our bacterial give results within 120 minutes without need of any human interference. Our competitor analysis, this competitor analysis based on Indian market, we modular innovation are our main primary competitors. Our kit give results within 120 minutes, but currently available conventional method give results after 20 to 48 hours. For doing current culture-based method, definitely need a skilled microbiologist, but our bacterial is a benchtop kit, so no need to very skilled operators. And another, our bacterial give real-time susceptibility results, but conventional method don't give these results. Then our kit is very affordable and benchtop. We plan to give our one bacterial kit for $500 and antibiotic panel for $10. For market analysis, in this market analysis, we analyze, we study two market. One study based on Bihar between 2020 to 2024 stated in between these 2 years, totally 40,000 tests taken in only 5 hospitals. If we targeted only Bihar, we can able to generate 86 lakhs per year. Then another study based on ICMR data, you see in 2021, totally 29 labs to 1,900 tests. But in 2023, 21 labs to 4,000 tests. You see, increase the number of antibiotic tests taken per year. It's our traction validation metrics. We do our clinical trial at Karpagam Medical College, Coimbatore. Our kit give more than 93% of accuracy. Then we give results within 120 minutes. We already filed one patent. We are working on another patent also. It's our professional patent file certificate. It's our current phase. Currently, we in early 2025 we start with urine-based analysis. End of this month we start blood-based analysis. In 2026 in Feb we plan to apply stethoscope clinical trial license, and then within the next year we plan to complete our final model. It's our team. I am Sumanth Murugaraj, CEO and co-founder. Dr. K.K. Sendil Kumar, he lead our research. Clement Selvam, he manage our fund flow. Sriram Shankaran, our mentor. Dr. Deepak Raghun, he handle our UT-based clinical trials. Dr. Vidya, he handle our synthetic receptor development. Arun, handle our hardware development. Our ask, we need to hospital support for doing our clinical, preclinical and clinical trials at various geographical wise. Thank you so much.
Fantastic, Sugandh, completing it within 4.5, 5 minutes. So great job. Some time for question and answers now. Mandeep.
Sugandh, I just want to say I have a lot of questions, so maybe we should take it offline. But one of the main questions I will share here is I would like to really understand the reliability and validity of the methodology, because I think that's going to be very critical to understand how the other pieces. I understand I tried to look very closely with your confirmatory study piece, so I would like to look at even closely because I couldn't see that even I tried to zoom in. But I will say I know it started with the bacteria, but then going through that whole process, my questions are more about reliability, validity, and then also the narrative was 60 minutes because you get the sample in results in 30 minutes. Sorry, 30 minutes?
120 minutes.
Yeah, but then one slide said your 2 results are happening in 30 minutes first, and the second one is also 30 minutes. So that adds to 60. So I want to know the difference between 60 and 120. I think that'll be nice to understand. And then also one question was around market analysis, analysis that you showed, because you also mentioned that the reason you started doing this was because you want to provide support to small hospitals who don't have this methodology. So, but then the market analysis were more towards the bigger regions and hospitals, right? So just kind of connecting the dots. That's why I said, more questions, we can take it offline, Harsha, in the interest of time.
Absolutely.
Actually we start with our co-founder, doctor affected with multi-drug resistant anti-infection in 4 years before. So we start this project. Our methodology based on membrane potential. We take the bacterial sample and load it into the antibiotic chamber. Then we apply one signal to the chamber. The sample have bacteria. We got one another signal from bacteria due to ion efflux. But if the antibiotic effectively kill the bacteria, we don't get any signals from the chamber. Based on that, we differentiate the sample in antibiotic effectively kill the bacteria or not. It's our basic methodology.
Yeah. So we can take this further offline as well. Thank you, Mandeep. Chidi, if you have a question, please raise your hand. I have Christian's hand up, so please go ahead.
I really like this tech, and I would ask you to start thinking about what happens next. Let's say you commercialize this, or you go after your process. It's working, and how do you scale this? And the thing about selling into diagnostics is hospitals are buying supply chains and service levels and execution. And so I think about like the next part of the story to tell investors is like, who's going to acquire you? Because I think that's your natural path here. And, how long is that going to take you? Because in today's world, therapeutics are having a hard time, but these diagnostics can move quick. And it looks like you have something that is novel. So, one, who are the 5 strategics that you're going to get into? If I'm thinking US and how it works here, so it may not translate well to your market, but who are the 5 customers you're pursuing? Go get 2 of them. And then who's going to buy you, and go start working with those 3 and show them that you have a better product than they have. Show them that, and then prove it with your strategics that you're working with. And then you can look at the market analysis and say, this is what it's worth, and take some sort of license or sell it outright. But I like this. I think it's all about strategy and also traction. That strategic is so much more important than a regional hospital that nobody's heard of, right? Getting into Mayo Clinic's CLIA labs and saying, hey, Mayo, you can rinse and repeat and be twice as profitable on these tests. And they will, anyway, I love the product. Good job. Thank you.
Thank you. We complete our UTA-based infection trial. Now we are moved with bloodstream infection trials. Then already we got one interest from take our technology. Already one company interested to take our technology completely. But we are working on that. We complete our UTA last month. We start the bloodstream infection this end of this month. It take another 4 to 5 months for complete study.
Excellent. Chidi, please go ahead.
Christian stole all that I was gonna say, but I really like this technology as well. I believe that there has to just be a lot of strategic inputs into the scalability. But this is definitely going to sell. So yeah.
Excellent.
Actually, we are working on that. Actually, I shared the data based on Indian market, but we also analyzed African and the UK market. There is more need in African US market. Then most of our completed technology based on DNA props and other fluorescent enzymes, but our method purely based on reagent free, like fluorescent or dyes free, and purely depends on cell membrane potential. So
Okay.
it support wide variety of organisms.
Great. Abhishek, did you have any quick comment or question?
Yeah, just a quick comment actually. Sagant, I think what would really help is if you talk to potential customers to understand the positioning and explain the gap that you are trying to mitigate, and understand the key nuances of the features that you want to have in this system, right? Product is great, but as Christian also called out, the scalability and positioning to the customers, I think that is very critical. Thank you.
Thank you. Thank you.
Wonderful. We are up on time. So thank you, Sugant. Great job. We'll move on to the next pitch. Judges, please take the time to wrap up the scores. Next up will be Dr. Undurti N. Das from EFA Sciences. Please take it over, Dr. Das.
Yeah. Okay.
Yeah, yes, we can see and hear you now.
Okay. Thank you very much. I'm a physician scientist working in the area of lipids as potential drugs. And in the current market, if you look at all the drugs are based on, monoclonal antibodies, synthetic chemicals, and so on and so forth. There are no drugs that are based on lipids. So lipids are a very important material in the body, and they form the component of the cell membrane. And we think that these lipids have been neglected. And so we have been working in this area for the last 30 years, lipids and their role in various diseases. One of the areas where we are currently working is in the treatment of glioma. Glioblastoma multiforme is a very lethal condition, and there is no cure or there is no appropriate treatment at the moment. And
would you like to go full screen presentation mode?
Yeah. Is that clear now?
Yes.
Yeah. And glioma is considered as a orphan drug status because there are only 18,000 cases of glioblastoma in USA. And in India, the likelihood of the incidence is higher. And generally, it is treated with debulking surgery, where the neurosurgeon tries to remove the mass of the tumor, but he cannot remove 100%. So at least 10 to 20% of the tumor is left, and it very rapidly recurs. And then subsequently, the patient undergoes radiation, chemotherapy. With all these therapies, the survival is not more than 1 year. So most of the patients, once they are diagnosed with glioma, they die within 1 year. And the people who got the disease and died within 1 year are Ted Kennedy and Biden's son. And this is an orphan drug status. The market value is about $0.5 billion in USA alone. And because it's an orphan drug status, it will have a FDA fast track and then reduce the trials of cost. What we did is we did a lot of in vitro studies and animal studies, and we have shown that our molecule called as gamma linolenic acid can very selectively kill the tumor cells without harming the normal cells. And there are no systemic toxicity and no local toxicity. And this is a slide showing the patient treated before and after, and it reduces almost 90% of the tumor, and whatever little tumor cells that are remaining, they are all dead cells, and so they do not recur. And we have patient follow-up for more than 1.5 to 2 years, and some patients even followed up for 3 years. There is no recurrence at all, and it can be done as an outpatient procedure. And the mechanism by which it occurs is it accumulates in the tumor cells and produces lipid peroxidation. Lipid peroxides are very toxic, and it doesn't accumulate in the normal cells, and so there is no toxicity to the normal cells. And this seems to be the natural mechanism by which the body eliminates the tumor cells and unwanted cells. And this kills these lipid peroxides kill the tumor cells by a process called as apoptosis. And here what the method of administration is directly we inject the GLE into the tumor bed, and then every day 1 milligram is injected for about 1 week to 10 days. And by the end of 10 days, no more injections are necessary, and the tumor regresses, and there is no recurrence. And we have already done preclinical studies in mice, rats, and rabbits, and there is significant tumor reduction, and there is no brain damage by histopathology and by EEG and other methods. And we have already done an open label study in 21 patients, and 1 milligram per day for 7 days it was given. The patients were alive for more than 2.5 years. There is no recurrence, and there is no side effects, and this can be done as an outpatient procedure. The catheter is inserted at the time of debulking surgery, which is a must as per FDA guidelines. At that time, they put what is called as a Ommaya reservoir into the tumor bed, and we can inject the drug into the reservoir and then press it, and so the drug goes into the tumor bed, and then the tumor is completely regressive. So this will be the treatment for glioblastoma multiforme, and current treatments will not produce survival beyond 1 year, whereas our treatment produces more than 2.5 to 3 years. And the pricing, we expect that currently it costs about $2 to $3 million. And whereas we try to be as reasonable as possible, each injection probably costs more than 50 to 70,000. And so it will be a license to a global pharma if that is possible. But it is also possible that we want to have a standalone company because we have other lipid molecules under development for various other diseases. For example, we have a lipid molecule under development for diabetic retinopathy. And for diabetes itself, it prevents both type 1 and type 2 diabetes. And for uveitis, which is an autoimmune eye condition, and also for other conditions we are developing. So they are in different stages of development. And we compared with the current modes of treatment. All modes of current treatments are recurrence is extremely common, and there is no cure for this disease. Whereas our drug gives a complete cure almost, and there is no recurrence at all. And the advantage is the longer survival, no side effects, and then it is cost effective. And we are now planning to do the GMP manufacturing, and then we will go for the FDA approval to do the clinical trials. And the clinical trial would be start from the phase 2, because phase 1 is not done because it cannot be injected into the normal patients, normal persons. And so we'll directly go to the phase 2 study, and then we expect it to be completed within 1 to 2 years. And because it is an orphan drug status, it will be approved very fast. And there will be, then we will try to license it to other pharma and then go for other drugs.
6
And
minutes warning.
yeah, and because it's an orphan drug, we'll get 7 years exclusive marketing right in USA and 10 years in the EU. And I'm an expert in lipids. I have published more than 400 research papers, and I have more than 5 patents in the US and others. And then we are now currently asking for about 2 to 3 million for going to the GMP manufacturing and do the clinical trials. And it's a safe, targeted therapy and life extension, and absolutely no side effects. Thank you very much.
Amazing. Thank you so much for a fantastic presentation. Judges, please unmute yourself as you have questions. Go ahead. I don't know, whoever, please be your own judge. Who is first? Unmute yourself.
I think Abhishek was first and then Christian.
Go ahead, Abhishek.
Okay. All right. A couple of questions. Great work on having this lipid-based drugs. So one question is around the manufacturing complexity. How would you control the particle sizes or polydispersity and charge distribution for this? Have you shown any data on a small scale? Second, the off-target toxicity in being the lipid-based drug, that's a major problem with them, and also the immunogenicity. How will you address the concerns? Have you done any formal study around it?
Yeah, manufacturing, we already initially we did all the preparation, everything in the lab and under sterile conditions, and now we have a manufacturer lined up to do the lipid based manufacturing, because none of the existing pharmaceutical companies are capable of doing any lipid based manufacturing. So we have now already found out one manufacturer who is going to do it, who is a known lipid supplier to all over the world. Number 2, coming to the charging and other things, which is a small molecule, their molecular weight is less than 350, and so very easy to handle. And it is a little bit unstable in the sense it has got 3 double bonds, but we cap it under nitrogen, and so there is no problem at all. And it is non-antigenic. So lipid being a lipid, it is non-antigenic, whereas compared to all other monoclonal antibodies and others, they're all antigenic. And so we don't have any problems at all.
Christiaan?
Okay. Thank you.
Okay, I'm just going to rattle off some things again. I don't know if there's enough time for back and forth, but just things to consider, Dr. Das. I went and researched this. It was really kind of hard to find you, but then when I found you, all kinds of great data about your career. So nice to meet you. Thanks for your contributions.
Thank you.
I didn't see much about past commercial successes and maybe regulatory expertise or these different areas that I care about as an investor. If I'm looking at the team that's going to be moving forward this early stage therapeutic, 95% failure opportunity, it's where it is. About 5 to 10% will succeed based on markets. So I really like to see someone in the lead spot who has commercialized, who has raised money, who has fought these battles before, and is actually maybe built into what's happening in the marketplace. So I'm just, that's my feedback. That being said, you may have that in your background. I didn't see it anywhere in your presentation, online, or any of those. Maybe I missed it. So I'm thinking like an investor hat, really cool tech. Who's going to lead it through this, gauntlet that you have in front of you? Okay, thanks for listening.
Thank you very much. To answer your question, I'm not a commercial person. I'm a scientist and a physician. So I know that my drug works and that that it has no competition. And once we raise the $2 to $3 million, we are going to recruit a commercially capable person in the CEO, and then I would concentrate on developing other drugs. And we already had a pre-IND meeting with the FDA, and they liked our data, and they asked us to do one more preclinical toxicology study under GLP for where we injected in the rabbits for about 7 days, but they wanted it to be injected for 10 days and show that it is non-toxic. When we inject the
Dr.
drug,
Das, you diving in this way is kind of indicative of what I'm suggesting. You either have that angel funding that's going to get you there, but if you go out, there are many small molecule, incredible Alzheimer's, brain stuff, diabetes that are taking up family dollars. And I see the technical founder not getting funded today without a background of commercial success. And I'm just sharing with you, your success in the US will be much quicker if you have that person. And I trust you that your tech is the best in the world, unrivaled. You need a partner that can earn the trust of the market in that way at this time in the world where small molecule deals aren't getting funded. Okay. Thanks for listening.
You suggest somebody we are willing to take them and partner with them and go for the commercial angle.
Yeah, unfortunately, we have no more time. Sorry, Mahesh. Mandeep, please share your comments in chat, and we will definitely follow up with Dr. Das afterwards. Let's move to...
Thank you very much.
Appreciate it. Let's move to the last but not the least, Vihang Vivek Galsasri, IUVA Labs. Dr. Vihang, please take it away.
Hello, everyone. Good evening and good morning to all the panelists. Thank you, Harsha, for this great opportunity. Allow me to just share the screen and we will get it started.
Yeah.
Yeah, is my screen visible?
Yes, please go ahead.
Just a minute. I need to hide these. Okay, let's go ahead. Okay, again, good morning to all. Good evening to all. I'm representing Yuva Labs. We are located in IIT Bombay, incubated at IIT Bombay, and we're from Mumbai. We are a startup working at synthetic biology, recombinant protein expression, and precision fermentation. So before we start, did you know there are over 1 million Indians who have a rare genetic disease that makes them unable to process dietary protein? No. What exactly is it? We are talking about phenylketonuria. This is a genetic disorder, rare genetic disorder that affects 1 out of 15,000 Indians. How does this, what is the mechanism or pathology behind this? Phenylalanine is an amino acid that's naturally found in proteins. Generally, when we eat proteins, we digest the protein into amino acids, and the amino acids get metabolized into different metabolites, and that's how our body builds its different functions. However, in people who have phenylketonuria, this PAH enzyme, which stands for phenylalanine hydrolase, this enzyme is non-functional because of a genetic mutation, and affected people are not able to metabolize phenylalanine. Normally, this phenylalanine can get converted into tyrosine and then become a part of different metabolic processes. But because in the patients with PKU, this metabolism is impaired, this phenylalanine accumulates in the body. Eventually, it gets into the brain, and then it has neurotoxic effects. This accumulation leads to irreversible brain damage. So most often the babies who have this particular disease, they have retarded growth, physical as well as mental, and they have a very poor quality of life. The treatment options that we have currently are actually a free amino acid diet. Let's understand this a little better. There is a trade-off between palatability, efficacy, and cost. The one alternative that generally is being given to PKU patients is free amino acid formulas, which means there is no intact protein. They cannot eat anything that contains proteins from natural foods. So they are given a formula which contains all amino acids except for phenylalanine. However, this kind of a formula has very poor taste. That leads to poor compliance, especially among children. And it has suboptimal absorption, because if you give free amino acids in the gut, it saturates all the gut receptors and the amino acids get poorly absorbed. Moreover, it has a high cost, approximately 4,800 rupees per kg in India. There is a second alternative available, which is called glycomacropeptide. This is a natural peptide from casein protein that is found in milk. This is generally has a neutral taste and it has a better acceptance. It is superior than free amino acid formulas because it allows sustained release of different amino acids and allows for better absorption. However, it is extremely expensive, so approximately $700 to $800 per kg, and this is not made in India. This has to be imported. So there is an unmet need. There is an unmet need for having an excellent protein source which has good palatability, which is versatile, which can be converted into enjoyable food formats, and it has a optimal sustained release from a complete intact protein. And moreover, it can be manufactured locally in India. So what's our solution? The solution that we are presenting here is an engineered phenylalanine-free whey protein, which is going to be neutral in taste, so that it allows versatile and highly palatable medical foods. Something like it can be molded into a ready-to-drink beverage. It can be molded into different, just a powder, or it can be into a bar, or it can be into a spread. Different kinds of formats will have a high nutritional value similar to a whey protein that normally everybody consumes as a protein supplement. But then PKU patients can also benefit from it, and it will have excellent stability because generally whey protein is known for its ideal mouthfeel and stability, and this will also address the poor compliance that currently patients have. What is the approach that we are taking over here? This is a dual-pronged approach which combines AI-assisted design and a proven production platform. What we are going to do is we are going to use tools like AlphaFold and molecular dynamic simulation to exactly remove the phenylalanine residues that are present in the beta-lactoglobulin, which is a whey protein found in bovine milk. So we have already done a basic analysis. So overall protein of the beta-lactoglobulin protein has only 4 phenylalanine residues. And we have done a very preliminary comparison with different protein structures. And we see that these phenylalanine residues can be replaced by other equivalent amino acids, and an engineered version can be obtained, although it needs a lot of analysis in a computational format to check for its stability, folding capacity, and the protein should be able to express. So the current AI-assisted tools like AlphaFold will definitely be critical in coming across this barrier. Once we have identified some leads which are computationally stable, we will go ahead with gene synthesis and cloning, and we will express them in our high-yielding Pichia-Pastorius expression system. After the expression has been verified, we will take it forward for high-yield precision fermentation, wherein we leverage our optimized fermentation process to produce the engineered protein at scale, ensuring an efficient, reliable, and cost-effective supply.
5.5 minutes warning.
Sorry, 5 minutes done?
Okay. 5.5, yeah.
Okay, so the science is already de-risked. Our precision platform is already validated. We started in 2022 with, we have already worked on a wild-type beta-lactoglobulin, and the yield of this protein, we have improved over several generations of iterations of strain engineering, and we have already have 30 plus gram per liter of yield of our wild-type beta-lactoglobulin protein. And this has also led us to a significant reduction in the cost, starting with something like 80,000 rupees per kg. We have come down to 2,800 rupees kg with our wild-type beta-lactoglobulin. We have also confirmed its confirmed the molecular weight of the protein and the entire amino acid sequence by performing these analytical tests. And we already see that it is a 100% match, which validates our existing technology for the wild-type protein. There is a growing global market with a critical unmet need in India. The global PKU market stands at $600 million in 2024, and it is expected to grow to $1.1 billion by 2032. Currently, in India, there is no local manufacturing of PKU medical foods. All products are imported. The standard FAA formulas, as we already discussed, are at 4,500 rupees per kg, and the GMP alternatives are even more expensive. So there is a massive untapped need that exists for an affordable, domestically produced, high-quality solution. again, which will have superior palatability, superior nutrition, and superior economics. A little bit about our team. We have a molecular biology wing led by Mrs. Shama Bhatt. There's a strain engineering wing, which I look after. I hold a PhD in molecular biology and around 10 years of industry experience. There is a bioprocess department, which looks after the precision fermentation process. And another co-founder of ours, Mr. Saurav, he has a prior experience in starting and commercializing a food tech venture. So he looks after company fundraising, compliance, and other non-technical aspects. Our ask is rupees 36 crore, which is equivalent to $4 million. And we are going to utilize this money into hiring computational biologists, bioprocess, and downstream engineers. We are also going to scale up to approximately 10,000 liter fermenter scale and purchase lab equipment to increase the team output. So currently for this PKU formula, it is still at an ideation stage, but as I already explained, the wild-type beta-lactoglobulin technology has already been validated. We have already reached a stage where we have demonstrated it at the yields at the lab scale, and we are soon going to go forward to the scale-up fermentation systems. So the same platform will be utilized for this engineered beta-lactoglobulin variant, which will be used for PKU patients. That's it from my side. Thank you so much for your attention.
Great presentation, Vihang. Thank you. 8 minutes up, 2 minutes for Q&A. Mahesh, please go ahead.
Thank you, Harsha, and thank you, Vihang. Two questions. One is on the data that you presented. Is this clinically validated published studies, or is this kind of in-house? I didn't see any publications listed there. And then the second question is around the IP. Where are you with patents? Is it just in India? Is it global? Just walk me through a little bit more about how you're protecting yourself from IP perspective.
So the data that we showed is for a wild-type beta-lactoglobulin, like the one that is found in bovine milk, nature identical amino acid sequence, and that is an in-house produced data. This is not published anywhere. We are a startup. So we have in-house developed a strain. We have optimized its lab scale bioprocess, and we have done the mass spectrometry related analysis to confirm the identity of the protein, right? And we currently don't have any filed patents on these because the current product is a nature identical amino acid sequence. So as we say that the naturally found proteins cannot be patented, right? So that's why we have not filed patents on that. Regarding the PKU formula, the PKU idea that I've presented, we are at an ideation stage, right? So we still have to go ahead with this re-engineering of the protein and using computational tools. And the moment we find a stable amino acid sequence, which yields us a stable expression, that will be our IP.
Got it. So I know obviously natural products cannot have patents around it, but processes,
Right.
utility, all of that can. So it might be important to kind of think through, that protection there, because again, it's quite easy to kind of replicate
Sure.
from a scientific perspective.
Sure. So with respect to our natural beta-lactoglobulin as well, as we go up in the scale-up process, and we definitely expect to have some intellectual property arising out of it, because that is where the innovation will help us to meet cost effectiveness.
Abhishek and Christiaan.
Yeah. Hi, Vihang. Good to see you here.
Hello.
Quick
Abhishek.
question I
How are
have
you?
is how is this? Good. How is this product differentiated from like existing amino acid based or GMP based protein substitutes, right? And what's the differentiating factor, like taste or the protein content or the variety of protein content?
Especially biomarin.
Right. So the free amino acid formula is just a mixture of individual amino acids, right? You put alanine, valine, glycine, etc. And then these have bitter taste. They don't really taste well, and they don't get absorbed in your body that well, because when you eat, like when you ingest like a formula of amino acids, they saturate your receptors in the gut, and barely any of that gets absorbed, and then that gives you poor compliance. So understand one thing: PKU patients are not able to eat any natural foods, right? They can't eat your dal or roti or anything, because any natural food by default will have some proteins which contain phenylalanine, correct? So currently they have a very poor quality of life. Even if there are other therapies that might come to cure their disease, they still have to survive until that point of time, because this is something that is diagnosed at a very neonatal stage, right? So, and in very young stage, if the progression of this disease is not contained, then it goes into very severe neurodegenerative repercussions. And if mental retardation happens, then obviously it's a lost case. So it's very essential that since the infant stage, the patients are fed with good amount of palatable and easy to absorb, sustained release kind of food, which will help them to survive that stage and avoid any serious repercussions. So this is where we are trying to address this particular problem.
Last and final question or comment by Christiaan.
Awesome job on the presentation. I love your slides. I followed them very well. I think that where my head is more product market fit. What I'm seeing is that this is your beachhead, and you can do this probably for a number of different markets, right? And so it's who's going to buy this from you, and what did you do clinical work? Did you talk to the patients? You know, all that piece of it. If I'm going to invest, I think you said 5 million for basically production capabilities, and I have a bioreactor background, and I produced proteins, antibodies, not the same. But you know, who's going to be buying all this stuff that we're, you know, and I know that might be a ways off, but show me the voice of customer a little bit in this, and maybe
right.
just that.
So basically, the parents of babies who are born with PKU are our first targets, and the whole distribution will go through hospitals, especially nursing homes where young babies are delivered, right? That's where the disease is first diagnosed, right? The neonatal diagnosis, because generally there's a, yeah. Sorry,
I
you're saying
do
something?
understand that. I think I'm looking for you to tell me what the pull is rather than the push. Like I have a product, I'm pushing it into the existing stream versus, hey, I've socialized my technology and here are the groups that now want to partner with me to move it forward. That's where my head was. Like, is there a pull in the market someplace that you could talk about that gets me excited? And I'm like, oh, okay, he's going to go do it here and then he can do it all these other places. Anyway, that's my only feedback. I thought it was a very good presentation.
Thank you. Thank you. I definitely, we are a little early for that right now because this entire concept is at an ideation stage. And basically, I'm seeking feedback on this platform from you. So the point very well noted, Christiaan. I totally appreciate that.
Fantastic. Congratulations, Vihang, on a great presentation, and congratulations to all the pitchers today. Let's give them all a big round of applause. Fantastic job. And I will take a big round of applause for not keeping up time. So I maintain my record of never ending a meeting on time. Anyway, but great job, guys. And thanks to all the judges for fantastic questions. I know there's a number of ideas and connections on the chat. Please be sure to copy the chat before you exit this meeting because we will save it as well, but make sure you have what you need. And so if you are any comments or questions before we wrap up the meeting for everybody so I can have a brief debrief with the judges and close the meeting? Any final comments or questions, anyone? Okay. If not, thank you all for joining.